here to introduce Professor Eric Francis Wieschaus. Please.
Thank you very much, Okti. Just a very brief introduction, and thanks to all of participants who came with very interesting scientific presentations, and I think we will enjoy these more informal discussions also later. And I'm very honored to actually have now as a plenary speaker Eric, who was recognized actually with the Nobel Prize for his incredible findings about genetic regulation of morphogenesis. And today he promised he will give us some new directions or some new insights with a special lecture. So we are looking forward for that and discussion that we will have in the later also after the talks when we move back to the Institute. Thank you very much. And Eric, now please come and deliver your talk.
Can you hear me when I speak like this? Good. One of the most remarkable features of biology is that when I look at every one of us in this room and the great complexity of any human being, the realization that each of us can trace our existence back to a stage where we were a single cell, the product of a fertilized egg, and somehow in that single cell was the information that was necessary to generate the complex patterns that define who each of us are as an individual. Now, ultimately, this process obviously depends on genes and on proteins and the expression of specific genes in specific tissues. Neural tissue requires specific genes that are involved in neuronal transmission, muscle tissue for specific proteins that can generate contractile force. But the control of gene expression is not and can never be the whole story, because in what is our development, what is crucial is that these patterns of gene expression be developed in the correct spatial order. Our brains have to be in our heads, our muscles have to be in certain defined positions. So the basic problem in developmental biology, and it's the problem that I'm going to address today, is how do these patterns arise? How do patterns of gene activity arise during development?
It worked before.
So in many respects, this is as in most problems in biology, are questions of input, how input information is used to create characteristic reproducible outputs. In the cartoon that I've shown here, just describes a general pattern for embryonic development. and one that has been developed and accepted over the past 100 years. Development begins with the fertilization of a single egg cell. That egg cell then divides into multiple cells, and at some point during the process, individual cells begin to express specific genes. Now, the information for which genes are expressed actually in many organisms pre-exists the pattern of gene expression and pre-exists even fertilization, in that the egg, although it often appears very homogeneous, is in fact is graded distributions of specific substances, and those distributions, chemical distributions, provide input that ultimately control informational input that controls the output in terms of transcription. This is the model that most of us in biology use in thinking about development. What it requires is that the input, I'm sorry for this misspelling there, the input involves maternally supplied RNAs and proteins that supply spatial information, and the output somehow involves a response to that information, and specifically in terms of spatially patterned transcription in the embryo. Now, what I'm going to do today in my talk is actually organize the material in two basic questions centered on input and our studies on the nature of that information and the quality of that information. And the second part will be on output, how that information is actually used molecularly to produce complex patterns. The first part describing input will be based on experiments which we have largely already published or are in the process of publishing. The second part dealing with output and response are going to deal with very current, recent experiments that are directed at defining responses and understanding quantitatively how those responses occur. Now, the idea of input as a maternal structure in the egg can be dated back to more than 100 years. And the early embryologists who studied development characterized different molecular distributions in the egg that were inherited in patterns that suggested these cytoplasmic differences had impacts on development. More recently, and specifically in the Drosophila embryos, the fruit fly embryos that I'm showing you here, we know the molecular nature of those inputs. We also know that essentially they are graded distributions of molecules such that concentrations, thresholds define individual patterns of gene expression. Now, that previous simple diagram, as is often the case in biology, describes what's actually a somewhat more complex situation. In Drosophila, what we've learned over the past 25 to 30 years is that there are, in fact, 4, and I will argue only 4, molecules that supply input spatial information. That's an extraordinary simplification of the problem. But each of those 4 molecules is graded in a distribution along the anterior-posterior axis of the embryo or the dorsal-ventral axis of the embryo. Each of these proteins are transcription factors that bind to DNA and govern gene expression. Their quantitative distributions are reproducible from one embryo to the next. The specific gradients for each of these transcription factors are established by different cell biological mechanisms. Some of them are established simply by localizing an RNA. Others are established by a graded control of translation or by cell signaling complexes that govern either protein stability or nuclear localization. So there are 4 systems here, and all of these proteins are supplied by the mother, built into the egg during oogenesis, and involve gene activity in the mother. When those genes are mutated, the mothers develop perfectly normally because they came from mothers, if they came from mothers who were themselves not mutant, but a mutant female will produce abnormal eggs where one of these components is missing, and the embryo will develop in corresponding patterns that indicate the role of that gene. It's important to emphasize that this isn't the only contribution of the mother to development. She provides these 4 information molecules, but she also provides virtually all of the RNAs and proteins that the early embryo needs for its survival. So there are about 6 or more than 6,000 different proteins in the egg. But in contrast to these spatial cues, these proteins, to the extent that they have been analyzed, are always uniformly expressed, and they're needed in all cells, or at least are available to all cells, and they don't supply information about position. So the central and really important advance that has been made in the Drosophila genetics research has been to reduce the input information down to 4 measurable gradients, each of which supplies quantitative information for further development. Now, that's an aggressive statement that these are only 4, and one can ask why. How sure are we that these are the only information inputs? One strategy, often, or that information is based on the fact that when we do genetics and I try to identify mutations, one makes more and more mutations, but the only thing that one finds are mutations in these genes, only mutations that change the pattern of the embryo. From a maternal standpoint, are mutations in these genes or in other genes that are involved in controlling the spatial localization of these 4 products. So that's an argument that we've looked and looked and looked and can't find any others. But there's a stronger argument, actually. One that I undertook a couple of years ago was to ask if these are the only information that the embryo uses to establish pattern, then if you eliminated any one, you might get a complex remaining pattern. But if you eliminated all of them, all pattern should be lost in the embryo, and the embryo should be totally uniform. And gratifyingly, a strategy there is to make quadruple or double mutants. And gratifying, what one sees is that instead of, in terms of patterns of gene activity, which prefigure future subsequent development, as shown here in a wild-type embryo, when you eliminate these 4 genes, the embryos that are made, in terms of every gene product that we've looked at, are now uniformly expressed throughout the whole embryo. And so that tells us that there is no underlying undetected pattern or underlying undetected biases or spatial information in the embryo, these 4 genes have to be and are the only input. And I will emphasize that in biology, it's very rare that we have systems where you can define an input so precisely. Now, we can make the experiments more precise by not only by not simply removing these genes, but instead flattening all of the gradients such that all 4 of these systems are set at a single level, but we can adjust the levels of those proteins up and down. And what that allows us to do is to mimic different positions in the embryo, different words of the information code, such that we can now, if the information code defines a certain combination of these gene products would define nervous tissue or muscles in the arm, we can now program the entire embryo to a single fate, and we can shift that fate by shifting the relative levels of these 4 products. Now, one of the, this now we believe puts us in a position where we can actually understand the biological, molecular, chemical processes involved in providing information and then also using it in terms of transcription. One of the interesting experiments is once you know these are the only 4 systems that are required for patterning, and that the patterning is totally absent when you remove all 4, you can add back any one of these systems on its own and ask, how much information does this particular system provide? So I'm showing you an experiment here for one of these factors, a factor called picoid, a homeodomain transcription factor. And the critical idea here is that if one makes, this is the normal product of embryonic development, it's a little caterpillar or a little larva. After 22 hours of development, that hatches out of the egg. And what we see here is that if the only information that you have is from one of the molecules, picoid, you get a substantial part of the pattern, but certain parts are missing. If you then, from this embryo, if you now remove that single factor, you get an unpatterned embryo. We can measure the distributions of the protein, we can measure the responses, and a remarkable feature is that this gradient of the picoid protein can provide threshold information for expressing all the genes in the pattern of the embryo along the entire axis. Now, we've done this kind of experiment for all 4 systems. And what emerges is an interesting observation, namely that each of the systems is capable of patterning more of the embryo than what is missing when you remove that system in an otherwise wild-type embryo. That is, each one of these systems provides substantial overlapping information for the patterning. The consequence of that is that individual, there are pattern elements, parts of the pattern, which can be formed by any of the inputs. There are other pattern elements which can be only formed by a single input. And so we'll be able to begin to sort out the information. The pattern elements can be supplied or the information can be used from any of the system, argues for a certain redundancy in the system, that a certain different informational states can yield the same final outcome. It's interesting, and a fundamental question in biology is the existence of such redundancies. How is it that organisms, how is it that cells, how is it that nuclei use multiple information sources to control the same outcome? What we believe in Drosophila is that these information sources are constantly evolving and changing over time, that of the three systems that are involved in anterior-posterior patterning, for example, one of them is very recent and has recently evolved in the history of Drosophila, and others exist in some form or other in broadly related animal species and therefore can be traced back to earlier stages, earlier points in evolution, and that they're shared broadly across many different organisms. What we've been able to do, since we understand these systems, is to actually, at the evolutionary level, evaluate how much information is provided by the new systems and how much is provided by the old systems. And what we found, and to do that approach, and these are experiments done with Bill Bialek and Bauer, is that to measure information, one can measure the gradient itself, but the really relevant question is the extent that the information is reproducibly used to establish patterns of gene activity. So you can evaluate information in this approach by evaluating the reproducibility of the response to the information, how variable the gene expression patterns are. And we know that in wild-type embryos, which is shown here, over a substantial fraction of the embryo, somewhere between the variability, the error, something that on the order of about 1% to 2% of the entire position. Since there are about 80 cells along the anterior to posterior axis of the embryo, this level of precision at 1% to 2% means that in wild type, the information, the total information from all of the system is enough to make each cell different from its immediate neighbor to the anterior or posterior. Because reproducibly, the embryo is able to achieve this low level of error. Now, we can go back and we can, instead of looking at wild-type embryos that have all four systems, we can ask for each individual system, what's the error rate? And what we find is that for substantial regions of the embryo, the error rates of the most recent, the evolved system, are comparable to wild type. And whereas other systems have error rates that are at least double to triple. And so what that suggests to us is that in terms of usage of information, even if all of these systems have the same information, the animal is using the most recently evolved information in making cell choices, or puts a substantial effort. And there's virtually no difference between the decisions in this region of the embryo, the decisions that cells make when they have only one input versus four in terms of their reproducibility. So we think this generalized approach for measuring input usage in terms of reproducibility of output is a powerful tool to dissect how information moves through the system, but then also a tool for characterizing how information evolves over the course of evolution. So, what I've told you so far is in an analysis of input that we are defining input as information from from maternal RNAs and proteins. That information depends on spatial localizations of those proteins. It's graded and quantitative, and it's combinatorial, but I will argue is actually, from an analytical standpoint, analyzable and potentially very simple. This is an exciting condition to be in. What we know, so at this point in the I'd like to just move on to outputs and responses. And the lesson here is that the output responses are probably more complex and probably involve a variety of different levels of usage of that information. With one part, what one would like to know is, are outputs strictly transcriptional? Is that the only path through which information is transferred? Are some steps in the differentiation of cells not transcriptionally controlled, or at least not directly the product of differential expression of gene products? And then we would also we'd like to develop technologies for measuring differentiated states to allow us to compare one differentiated state with another. And at this point, I have to introduce a second set of collaborators on these experiments. This is a group in Martin Boer's lab at Princeton, and Martin is a world-recognized expert at the mass spectroscopy proteomics level. And what we've done is set up a collaboration to characterize not only at the RNA level, which is fairly straightforward, but at the protein level, does one define differences between different cell types, and how do those differences arise? And so it's that set of experiments that I'd like to present to you next. So one issue with the output and response is that ultimately the output are defined functional cells, muscle cells, nerve cells. And it turns out in biology that we actually don't have an absolutely solid sense of what the actual differences are at the protein or biochemical level between different cell types, between different completely differentiated cell types. So one of the first things that Martin and Argit Marishta and Cruz set out to do was to actually ask the question, how different are different cell types? We know that many gene products we can define as housekeeping. They're required for a whole variety of energy management for the cell structure. And often we think of differentiation as supplying on top of those standard functions additional activities. Transcription factors say that would define the cells as neuronal, and then also cell type specific products like neurotransmitters. So what Argit set out to do was to take from differentiated larvae, that is, at the completion of embryonic development, actually later as the larva gets larger, dissect out individual organ systems and carry out quantitative proteomics to ask how different are the two cell types. In this particular experiment, he's comparing a Drosophila kidney cell type called Malpighian tubules with an epithelial cell type called imaginal discs. And the surprising result was that the differences between these cell types is not limited just to a small number of transcription factors or gene-specific or tissue-specific gene products, that virtually the entire genome, the entire proteome, 87, 88% of all proteins, even proteins that are expressed in both cell types, are reproducibly expressed at different levels in one cell type versus another. And we find similar results whenever we compare tissues. This is gut tissue with neuronal tissue. Again, something like 89% of proteins are different, at least in their levels. Now, we can also ask, obviously, what are there proteins which are specific for particular protein types? And that turns out to be about 10% of the differences that we see. The vast number of reproducible differences, and therefore the vast challenge in development, is not to turn genes on or off, but to set them at specific levels that are characteristic and functional for that cell type. That complicates the whole question of development. It makes, in a way, embryonic development, particularly in fast-developing species like Drosophila, even more remarkable and mysterious, since entire development to functional organ, it takes 22 hours at room temperature, and one is able to not only direct cells into specific fates, but entirely transform the proteome over that period of time. So what one would like to do now is, rather than simply dissecting differentiated tissues like kidney or gut or brain or skin, and compare the differences, we would like to trace during the normal course of development, how are those differences established? And what's the time course? And the difficulty, of course, is though it's possible to dissect out and identify brain at final stages when the organism is well differentiated, it's becomes virtually impossible at earlier and earlier stages to come up with populations of cells that are on the process of differentiating brain or differentiating skin. So what we've done is try to develop an experimental system which allows us to do mass spectroscopy and proteomics analysis on multiple numbers of embryos, 50 to 100 in total, and without dissection, by using the genetic programming tools that we've developed that control input and develop genetic lines where all the cells in the embryo are directed to brain, all the cells are directed to gut or to Malpighian tubules or skin. And as you've probably guessed, those experiments involve genetic manipulations where we make fly lines that are where the individual input, all the maternal gradients are flattened, but the levels of the transcription factors are set to different levels such that now there is for example, a genotype that we'll call thoracic neurogenic ectoderm, a genotype that takes the input information characteristic of this region here, but now spreads it out over the whole embryo, such that the entire embryo could be directed to a particular fate. And there are, in theory, a whole variety of genetic conditions that we can develop. So far, we've only been able to develop 3 of these, and it's those, or 4 of these, and those are the experiments that I'm going to tell you about now involve comparing cells. These are designed to mimic either purely epithelial cells or neurogenic cells that are programmed to neurogenic fates, cells that are programmed to kidney or gut. Gratifyingly, once these lines are established, we can show that they are characterized by a characteristic pattern of responses. Any given gene that would be an output is either expressed uniformly over the whole surface of the embryo or not expressed at all. So a gene like brachyury expressed only at the posterior pole of the embryo would be expressed in the future gut cells, but not in the neurogenic cells. So simply based on the immunolocalizations, we've confirmed that the lines that we've developed make homogeneous embryos which have gene expression patterns that are characteristic of specific regions of the embryo that are fated for specific fates, for specific cell types. This is just another example. So the next question is, we can ask, how soon after programming does the embryo begin to change? Do these massive changes in proteomics occur by comparing a neurogenic line with a gut line, for example, in this experiment? And what we see is that within about 2 hours after the blastoderm stage, almost 50% of the proteome, 50% of the proteins that are expressed at the blastoderm stage have now achieved different levels in these two cell lines. What's been confusing and surprising for us is that even though we know that the initial response of cells to maternal input information is transcriptional, that cells decide to become gut or neurogenic ectoderm because of the expression of specific tissue type specific transcription factors, most of the proteins in the embryo are actually supplied by the mother and present already in all the cells immediately before the stage of active programming. And so this has become complicated. Then how does one adjust protein levels rapidly over? How does one adjust protein levels if the protein is already there and supplied? And how does one do that in a tissue-specific way? This has changed our sense of development to now focus more heavily on the possibility that proteins are, that a critical element of control is not just simply expressing, transcribing new genes, but instead controlling the protein stability or turnover of proteins. This is just to repeat to say that from our measurements, at least, I mean, there are 1,029, more than 1,000 proteins, whose levels are different between gut and neural cells, and but are actually there, supplied maternally, and must be, at least in part, regulated. Must in part regulate those maternal populations, we believe. So what that has led us to do is to begin a set of analyses to characterize turnover, to ask how much protein turnover there is during this process of development, and is it gene specific, or do specific proteins have altered turnover rates, degradation rates, and are these the specific behaviors of proteins and their stabilities dependent on cell-fate choices? Now, the global nature of that question requires that we develop strategies for measuring the half-lives of all detectable 6,000 proteins in the embryo over time. And that's the technology that I'm going to present to you next. It is a technology built on a technology that Martin Woer's lab has developed, and is essentially a labeling technology. Uses heavy water, H2O18, introduced into the embryo, which is very rapidly moves a label into the amino acid pools that are involved in making new proteins. And so by effectively a pulse or an introduction of this label, one can follow the degradation rates of all proteins that existed prior to that time, and therefore measure their lifetimes. And what we surprisingly find is that we had anticipated that most proteins would have, over the 22-hour period, would be relatively stable. Most proteins would be stable, and a small fraction potentially might turn over. What we find is that virtually all of the proteins in the fly embryo, 98% turnover, so turnover rates during embryonic development, that the average lifetimes are on the order of about 11 to 12 hours, and that they differ greatly. Individual proteins, there are proteins which have extremely short lifetimes and extremely and rather longer. But most proteins are turning over rapidly, and this gives the rapidly developing fly embryo the opportunity to establish distinct proteomes for specific cell types. The results with flies contrast with other organisms that we've looked at, even other embryos. So, for example, in Xenopus, or frogs, what one sees that about half of the proteins, we can't detect any degradation at all, or any turnover at all. Proteins are made, and over the course of embryonic development, which is much longer in frogs, proteins don't turn over. So one of the features of fly development seems to be incorporation of rapid turnover, rapid changes. Something that I can say as a developmental biologist is something that we've never really considered. We've tended to define developmental changes as changes in gene expression rather than as changes in protein degradation. So where we are now is that we're going back and beginning to do these labeling experiments on homogeneous populations of embryos to try to identify the cell type specific control associated with protein degradation. What would like to know is if there are massive changes, are there cohorts of proteins whose lifetimes are coordinately regulated? And if we can identify cohorts, then we're in a position to begin to look at generalizable mechanisms for how these cohorts of proteins are linked together, how one controls degradation rates, and whether there is a logic to how one groups proteins in degradation cohorts. We are not there at all yet, but that's the general direction, is to try to redefine developmental pathways, specifically in the sense of how one controls protein degradation. Now, I'd like to end this talk with a recent and rather surprising result, because it's been one that has equally transformed our analysis of development. As I mentioned in the beginning, the general view of input and output is that the mother in the egg supplies inputs in terms of small number of spatially distributed molecules, and cells respond and choose specific fates by measuring the levels of those transcription factors and activating genes in response to that. In that model, we make embryos that are where at the early blastoderm or blastula stages, all the cells have identical gene expression patterns, identical maternal components, and we believe are set out onto homogeneous, uniform pathways of cell fate. What we had initially planned was to then follow those embryos in terms of their gene expression patterns. An exciting observation, and one that we don't fully understand, is that even though the early stages are fully uniform, each of the genotypes that we've developed has the capacity to self-organize and establish distinct pools of gene expression that reflect specific cell types. So one of the first that can be visualized is we'll take, for example, the lateral neurogenic ectoderm, and in normal development, for example, there are genes which are normally expressed in stripes associated with specific segments. One of these genes is called runt. You can see in the normal wild-type embryo, this shows a characteristic pattern of expression, whereas if one flattens all the gradients, directs all the cells into the same state, expression of runt becomes uniform. Similarly, at later stages in development, about the final stages of these early decisions, the patterns are complex. So two different signaling molecules, wingless or Wnt molecule called wingless or in Grail, are expressed in stripes. To remove that, let's see. And rather than stripes, what you can see in the mutant case is that early on, these embryos are truly homogeneous at every level we can see. And yet, 2.5, 3 hours later, they've developed patterns, and patterns that in certain respects mimic the normal distributions of these signaling molecules in wild-type embryos, and that they occur adjacent to each other and are thought to self-enhance each other. So the embryo is So the embryo is able to establish or self-organize patterns that are essentially similar to the patterns that these cells would have developed, but they're doing this without any initial input. And so, not surprisingly, rather than the characteristic and reproducible patterns of expression that one sees in wild-type embryos, these are disorganized, and every embryo is distinct in terms of the pattern that is generated. So what we believe is happening here is that somehow the self-organization involves a capacity of the embryo to generate spatial patterns, possibly by signaling pathways that amplify stochastic variable differences in gene product, but then drive them in the nature of the signaling processes and the way the molecules interact with each other into patterns that then become stable and characteristic for that embryo, but different from one embryo to the other because the patterns of noise and the stochastic variabilities are different from one embryo to the next. And I'm stuck again here. I'm so sorry. We see this pattern in signaling molecules which are known to have positive and negative impacts on each other. Here you can just see the different, but perhaps more interesting, associated with these establishment of patterns, self-generated patterns of signaling molecules, one sees cell fate decisions that are made and distinct cell types arising in the embryos. The neurogenic region, classically in almost all embryos, gives rise to skin and neural tissue. In wild-type embryos, those distinctions and those decisions are related to patterns of gene expression early on. But if one eliminates those patterns, what we find is that the cells still are capable of generating those distinctions. They make neuroblasts and they make skin. The difference, again, is that rather than the characteristic and extremely reproducible patterns of individual neuroblasts or neurons in the nervous system of a wild-type embryo that you can see here in these red and yellow little dots here, this extraordinary pattern, reproducible and regular pattern. What you have here is a random assortment of different neurons. So what one lacks is the spatial pattern, the well-defined spatial pattern, but one has distinct cell types. And at a fine scale, one also can begin to observe specific spatial rules relating to how these different cell populations are established together. Other regions of the embryo, other uniform embryos are able to undergo this self-organization. So the posterior end of the embryo, which would be the proctolial internal organs, would normally give rise in a highly patterned way to future kidney cells and future gut cells. What happens when you make a uniform embryo from this region at the blastoderm stage? All the cells are the same, and yet again, the embryo is able to establish distinct populations. Now, a critical observation, or two critical, every genetic line that we have developed so far that is uniform at the blastoderm stage and was designed to mimic a specific region of the embryo, at later stages begins to undergo or is capable of self-organization and establishing different cell types. The second important observation is that these patterns, it's not as though everything goes crazy in the embryo and all different cell types are formed. The patterns and the self-organization that occurs in the neurogenic ectoderm is different and involves different rules than the self-organization that occurs in the proteome. One case you end up with kidney and gut, the other case you end up with neural cells and skin. So that raises the question of what is it that we are actually programming at the blastoderm stage? What is the information content of these cells? These cells respond to maternal gradients by establishing a cell type, but it clearly doesn't seem to be cell types the way we understand them, like neural cells or kidney cells or gut cells. Instead, the cell type that is established early in these embryos is a cell type that is directed towards specific fates, but multiple fates that can be generated through self-organization. What the normal embryo supplies to that self-organization fate, self-organization capacity, is a spatial pattern that biases the events biases the self-organization to give you a reproducible large-scale spatial pattern. But the individuality of differentiated cell types is something that's held at some level within each of the cell types. Obviously, when one sees spontaneous self-organized patterns arising in the embryo, it's very hard not to immediately think of Alan Turing and the models that he presented in the 1950s for self-organization. And these Turing patterns, which we know exist at a certain level or seem to exist, and this is on the skin of a pufferfish, have been identified, but very often and certainly in flies, we've never seen Turing patterns before and have not been able to, weren't even sure that cells would be possible to generate them. And yet what we see is that very possibly, I've come to believe, that virtually all living forms have the capacity to self-organize and establish patterns according to rules potentially that are relatively similar, but may not be exactly the same as Alan Turing proposed. But that in certain organisms, this capacity to self-organize is entrained and made more efficient by controlling initial inputs to drive you to patterns rapidly in the course of 4 to 5 hours to build embryos as rapidly as fly embryos go. Now, an interesting feature when one looks at all the animals on Earth is that you can, and look at the embryos, biologists have begun to kind of group them into 2 classes. Those which we sometimes call larval forms, where the embryos, the eggs are made and the mother egg is fertilized, but development happens outside the body of the mother. Development is rapid. Often the eggs are large, but there's no growth or increase in biological material. The whole idea is to take a frog egg or a fly egg and as rapidly as possible divide it into cells and turn those cells into functional cell types such that the embryo can hatch as a free living form, a tadpole or a larva or a tiny fish embryo, and develop, and then only then begin to take in nutrients and begin to grow. So that's one pattern. And interestingly, in those embryos, what one sees characteristically is a significant contribution of maternal information in the egg as the distributions, in terms of the distributions of RNAs and proteins that are already localized even before fertilization, even before there's an embryo. So larval systems that have to develop rapidly in this model would supply information, but they still retain the capacity for self-organization. The other class of embryos, mammals, provide the predominant example, and this would be human embryos as well, have very small embryos, develop extremely slowly. All of embryonic development in a fly embryo is completed before a human embryo even undergoes the first cell division. Extremely slowly, but with growth, constantly taking in nutrients because the embryo is developing inside the body of the mother. Under those conditions, initial patterning in the unfertilized egg may not be useful to provide inputs and biases to make patterns reproducible. And so those embryos, human embryos, are often thought and described in textbooks as being totally self-organizing, or at least self-organizing at a specific event early in development. We can see that development because it's possible from human embryos and mammalian embryos to develop embryonic cell lines that can be cultured, grown, and then clustered together, and they will self-organize into patterns of gene expression that are called gastroloids. So they are able, we know that mammalian cells have been able to self-organize. In a certain sense, the experiments that I've told you about on fly embryos are an attempt to transform fly embryos into mammalian embryos, or at least by eliminating all initial information, we have a system now where one can, with all the genetic and molecular tools available in Drosophila, to begin to study self-organization. And that was the optimistic message that I'd like to leave you with. I believe these embryos are still extraordinarily useful for proteomic studies because their fates are still restricted, but they will be more complex. So I end by pointing out that the work on maternal patterning has been largely collaborations with a number of colleagues in Princeton, specifically though those in the physics department, Thomas Greger, Bill Biala, Gaspar Tocek, and then Maria Petkova and Neda Harush, as well as molecular biologists in my own lab, Colleen Hannon and Shelby Blythe. The proteomics and this new attempt to redefine differentiation was carried out as a collaboration with the lab of Martin Bohr at Princeton. I'll stop there, and if we have time, take any questions. Thank you.
Eric, thank you very much. This was a fantastic lecture. I hope everybody enjoyed, and they should. And it is open now for discussion and questions. The other Eric has a question. Eric, please.
Eric, your question.
Outside. Maybe an ignorant question outside those problems. When you allow this protein by the mother, how does that actually done? It's biological secretion and fusion, and why that's it? Why doesn't the gradients change?
Okay, so that's actually an area that my wife has worked on more extensively than I have. We kind of divided the animal at fertilization, and I focused on everything that happened in the embryo, and she's focused on the way that patterns arise in this single cell, the oocyte. And I think I will summarize. The oocyte is made in the female, and in Drosophila, the development of the egg is a multicellular process. There are many cell types in the ovary, and to a certain extent, the patterning and localization of factors depend on the overlying cells that, in a certain sense, impose pattern and impose localizations inside this single developing oocyte. But what has been very interesting in much of her work is the realization that it's actually a communication process between the oocyte and the follicle cell. So what happens back and forth is through this communication, which involves well-characterized signaling molecules like EGF, an EGF receptor, or BMPs, or any of these signaling molecules, the mother, the cells of the ovary, are able to establish within this single egg cell a complex pattern. And that complex pattern is stable. Once the egg is finished, it can be held in that pattern until fertilization. And then that oogenesis pattern, to a certain extent, is released and evolved. So some of the molecules, that fertilization elicits translation of localized RNAs to produce gradients. It also triggers the release of certain chemical signaling molecules that are localized in specific places, actually in the eggshells themselves, to elicit responses. And so I suppose what one would say is that reproducible patterns in life and in species where pattern is always built on previously existing patterns. So the pattern of the egg is built on a pattern in the ovary, and the patterns of the follicle in the ovary is built on a pattern of stem cell divisions in the top of the ovary that are giving rise to I can see he's getting a little uncertain of my simplification, and that in some, so one can trace back through every generation, the patterns are maintained. The unpatterned embryos that I've described to you here are clearly dead ends. The normal development, I believe, and the normal survival of a species depends on this continued maintenance of pattern, pre-pattern, pre-pattern, building on previously existing patterns. I think that's the general strategy for reproducibility. Thank you. Please.
The first axis that is established in the science is sort of an anterior posterior vegetable, animal vegetable organization of microtubules. And so you get the first sort shape of some elements, two different holes, which then get, as I was saying, translated translations triggered by the that's really good. That's right. in the flies, actually, what we then need is a separate, I mean, all the coordinate system of a 3-dimensional A. Basically, we need 2 coordinates to define each of these cells. And so in flies, actually, I'd like to add that there is an act of plasticity whereby the orthogonal nucleus, which is initially positioned in the center of the oocyte, receives, we don't quite understand the transdisciplinarity of the pathways, but it's also tethered by, but there is from the outside at a certain point in time an input that's released that inputs travel to the periphery, and where it travels seems to be stochastic. It seems to be dependent on maybe little shading inside the passing. That's a big part of it. We don't achieve the passing. And if you look at this, two things that I really go around and we have things that get established. So it's very fascinating.
There are many levels of this kind of molecular recognitions, which I suppose are not known exactly how you. So these are enormous areas still to embrace. But what is fascinating for me, and I will come to the question also further, that you have this circular kind of the morphogenesis. So that means you have to have some kind of morphogenesis for the oocyte, which will determine the pattern. So that brings us to this evolutionary question, of the multicellularity, and how this is established. These cells would have initially to recognize each other. I'm working on cell recognition, obviously, on different levels. So, and do you have any comment of that, or maybe we can discuss it later?
Right.
But anyway, I think this is a fascinating thing how this occurs, because you have these morphogenetic processes cycle at a different level, embryonic and adult kind of development.
I think a great unexplored area, which probably touches every aspect of developmental biology, from oogenesis to early embryonic patterning and signaling, to the movements of cells, to the restructuring and morphogenesis, in multicellular organisms, is the surface contacts between one cell and the next. Signaling molecules are on the surface, and we often think of them as just information transfer, but they all involve physical binding, and therefore the generation of physical forces that at some level or other could easily play out in subsequent events. I think one of the exciting things about developmental biology and the extent that our knowledge has become so deep about embryos is that they have become now one of the prime systems, or maybe the prime system, for understanding cell biological questions. Because embryos, particularly fly embryos, frog embryos, are easily available. They can be engineered to be relatively simple, but yet functionally relevant, where specific aspects can be assayed. And so much of what we know about adhesion, and historically, this also from the vet, involves work on embryos, and that's going to be more and more true. There was a question in the
still science. And then for a while. What kind of the way the
The room is echoing very, very different. Would you help me with the?
Yes. Okay. The question was actually, like you see in your images, the empty spaces or so. But these are, as I understand, they are not really empty spaces, just showing the pattern of certain gene expression. But you can.
Okay.
So it was that.
So
what is empty, right?
What no, what is empty and what is full is a hard question to answer. What is certainly true is that there's always water, and that water is not just water. It's generally a colloid gel, a concentration of molecules. There is structure there. In the fly embryo in particular, the inner volume, the cells are on the surface, and the inner volume is full of yolk and other proteinaceous material. There's nuclei there. We see, but have not investigated the role. No one has ever figured out how to eliminate the interior and then ask how the cells behave. Because you eliminate the nutrient sources at the same time. We do know that if you put a nucleus into the center, into this yolk mass, and subject that nucleus to the same developmental cue, the nucleus responds by transcription of patterns. So we can take this inner mass, which is biologically, I believe, only relevant for nutrition, and it is also patterned. And I told you what my belief was, that this is no information, boring. We're focusing on the cells on the surface, but we don't really know that. We don't know if there are roles, additional roles. And the way a geneticist would approach that problem, you've probably figured out, is to somehow alter the gene, do something to change things and ask, is there a consequence? That's the great power of genetics. It is not clear to me whether that's an approach that could be used for these apparent empty spaces in the embryo. Yes, please.
This kind of investigation was that's why which you really by self-composition by feeding the water by this maybe it's possible that this dynamic I believe any truth for which you see something or maybe measure something is potentially powerful. It's a various strategies are useful, particularly useful at the level of intracellular organization. And intracellular organization could be an egg, which is big, but it could also be individual single cells. And the behavior of single cells could be parallel the behavior or the behavior of single cells. Here, what they mean, how they behave. It is possible now. And you see what we were doing 40 years ago, we used immunoplanting for a specific protein, which uses recipe related variation of the human factor. That's not the same kind of localization of information, different non-invasive technology that therefore has more whether the spatial organs. The question is, when it said was the analysis has been scaled up, therefore the leading celebrates or characterized. If we find it in use, how do we think of the technology with my preference as a scientist is to use the simplest technology that is available. It gets me to the question that I want to ask. That means that I am really learning other technologies that I have available. They're still beautifully useful. So the challenge would be using technology and see something that I've never seen before, or that I never expected to use this. Then I can see always the exciting future of media. Any other evidence? Thank you very much for tonight. When we got all of this, it's nice to be going to the wall. Then stay closer if I'm getting these words in words. I guess for us, we talked about the mentioned one point the coordination by I think it's an old question that you people also be what extent is the transcript coming information providing information of the program that was going on, what is that person? The question is a different thing with the Mandalos proteins and different and as soon as I don't get much based in similar and then it's very tough. I think the question is when you characterize proteins, when you characterize RNA. And the RNA technology teacher, I want to the advanced editor is taking up available in single cell sequencing characterized protein binding. What I believe once is that would be someone is there's always a discrepancy between protein and RNA. Partially because RNAs are shortly related proteins are generally more related. And so proteins accumulate unless you forward it with RNAs, even the one with proteins have shortcomings. They don't do that. The 5 is the RNAs and we can like that. So there's going to be a discrepancy. But it's a peculiar thing that's Germany, that happens in Hungary. If the mother makes it in egg, she puts in RNAs and also the physical proteins, but also RNAs and peculiar statement. And how much protein comes from those RNAs is very disruptive, very difficult. I think RNA protein levels will be much bigger in embryos than in, say, in cell type, culture cells, or in cancer cells, where you still see differences. But I think characteristically, really consistent. In early developmental stages, very largely full of RNAs, with small number of together. There's more of it. I think people how we have focused on protein. It's not just about the last proteins, but also I can see the US also particularly women.
So I was wondering if this technical development can we explore those possible activities to make that small example that we need the strong scientists.
Yes, not just us, other labs are planning this stuff. In particular, there's certainly signaling happening. The central players dependency. The clearly very unique is continuing. But in the surface, we phosphorylation. We know that exists. One of the features of phosphorylation, what means it seems to be really actually it's and everybody is like I can't get preparations and temperature. Me personally, and also I'm not sure we should realize that sophistication we are one of the complicated traits is the post-reactivity of most of the kinases. And therefore the populations approaches usually possibly in response to an activation. Sodium, which is the relevant response. I know that very seriously, but I don't think they will be easy. Phosphorylation is really already major tool that I understand. And yet, for some reason, you could phosphorylate maybe proteins that may be highly effective at one super small number of the targets. It's certainly not how that works. It's a great challenge. So many different I think having mentioned it for the event as we we will continue that how meeting we try to follow a lot of discussion, not getting to follow exactly because it's time to the Bible. And now later we don't consider it is basically stuff. And we use what we get more deeper than we actually. Now I usually the next speaker. And we have a slight change in the program. I feel the coming up to the speaker because one of the colleagues that we delayed the plan. So please.
Thank you very much. No, it's not. Asta pe București. Nu se deschide? București? Ok. So good morning to everyone. Oh, I'm too loud. So of course, I want to thank very much to Eugen for inviting us and for making such a wonderful conference. It's a pleasure and an honor to talk today because what is more beautiful to talk about what you like more to do in life, playing with microRNAs. And also an honor because this is the most famous home of science in Romania, Romanian Academy. And of course, it's wonderful to talk after Dr. Wieschaus on something that I